Great. Well, I wanted just to spend a few minutes kicking us off, and I know we have a great group here that is very involved with eSTAR, but today is our eSTAR annual meeting broken into individual segments. So today we are focused on TGX-DDI. Before we get into the bulk of the TGX-DDI updates, I wanted to just welcome everyone. As I mentioned, I think most of you here are very familiar with HESI, but this is the Emerging Systems Toxicology for the Assessment of Risk, otherwise known as eSTAR Committee. And as many of you know, HESI is a scientific nonprofit whose mission is to bring together scientists from academia, industry, and government, and NGOs. We have a really nice mix on the call today and on this TGX-DDI team. The eSTAR Committee's mission really nicely is kind of focused around our genomics projects, but we think a lot about how to deliver systems toxicology approaches for risk assessment, catalyzing adoption of new translational and predictive tools, which you'll hear more about today, and even more so thinking about biomarkers for toxicology. So TGX-DDI fits really nicely into this portfolio. And within the eSTAR team, we have a very supportive public sector participants and private sector participants. Just wanted to put them up on the screen to say thank you for your commitment. And under our private sector participants, we have our members listed here, but also just want to note that we have additional company support through one of our projects. Our OASIS project has some tech partners, but for today's call, we'll hear about some of the companies that supported our ring trial. So again, thank you to all the support for our committee. The eSTAR committee has 6 working group projects. Today we'll hear about TGX-DDI, but if you're interested in any of these others, please let us know. Connie and I are in the midst of having annual calls for all of these. So if you missed any, we have them recorded and we do have some coming up. And just to wrap up my little eSTAR segment here, I just wanted to note that our committee has put out a really nice series of publications this year. So there's 3 listed here that were published pretty recently. And today we'll hear a lot more about the TGX-DDI ring trial paper. And the last slide here is just to note that we will be at SOT in San Diego in March and the ICEM conference in Edinburgh in September, both showing eSTAR projects. If you'll be there, we would love to meet with you. There's still time, I believe, for late breaking abstracts too. So if anyone has ideas, please reach out to Connie and I. We're always happy to put together some sessions, and we would love to see you while we're there. So today we're hearing about the TGX-DDI journey, and I would love for participation from the group. So please feel free to type in the chat, to raise your hand, to react. As we move through, we don't need it to be a super formal presentation, but just to hear from the group would be really lovely. Okay, so today we're here all to talk about TGX-DDI. And I'd like you to take just a special note of the title of this presentation, because today I hope to update you on some of the really great progress that this team has made, but also to emphasize the long journey that we've been on and how these achievements have come over many years of a lot of persistence and perseverance from the team, and just to kind of showcase that many years have gone by, but a lot of progress has been made. So I'll start with just a few key achievements. We will hear a history lesson from Professor Carol Yock, and we will have a presentation of the TGX-DDI ring trial that was completed within the last 2 years. Then an update on the FDA qualification, followed by a presentation from Andrew Williams on some updated analyses for the TGX-DDI tool. And then we'll have a time for presentation and discussion of some ideas moving us into the future. So really nice packed schedule. I'd like to get us started, but not without starting with my acknowledgement slide here, where again, a very persistent team. And I know there's been more people over the years that have been on this slide, but just wanted to acknowledge the ring trial team that will be hearing a lot of the results from today. So 2025 has been marked by really, really huge successes. So as I mentioned in an earlier slide, we have a TGX-DDI biomarker study that you'll hear the results from Vicky today, but this had been started a few years ago with design, and it's been implemented. It took time to run the study, to do the analyses, to write up the paper, and as you can see here, it was published in ToxSci this year. We've also spent a good amount of time submitting an FDA biomarker qualification package, and I will give an update on that after the ring trial. All right, I'll pass it to Carol to give us a refresher on where we've been.
Thank you very much. So this is a monumental year. We've been on this roller coaster ride and we've persevered through it. And I've been charged with telling you a little bit about how we got here. And, you know, this project started before I joined the committee. And I see people like Al, Yuri, Hydran, Patricia, Roland are on, Cyril. So if I get any of this wrong, you guys who've been here for a very long time, or if you want to jump in, just go for it. So the journey began about 15 years ago well, prior to that, there was, of course, the discovery of the biomarker. But in 2010, in terms of interaction with the FDA, that was when the first letter of intent was submitted to proceed with the biomarker qualification plan under their biomarker qualification program. And, you know, a lot of times people ask me, well, why on earth did this take so long? And you'll see from this history lesson why that is. The challenge was, well, those of us who met in 2011 with the FDA developed a qualification plan and proceeded through this full qualification plan led at Georgetown University by Heng-Hong Lee, looking at a set of 42 compounds selected in careful collaboration with the U.S. Food and Drug Administration to cover a whole bunch of mechanisms of action and really show the range of application of TGX-DDI for this validation process. And it was really, really successful. And in fact, the paper was eventually published in PNAS in 2017. And in 2016, the FDA reviewed the data and invited the full submission of the biomarker qualification package. So we were really excited at that point. And the team put together a package. There weren't really formal guidelines on how to there was no template or anything for how to put this package together. And we submitted something that we sort of jokingly referred to as a gigantic PhD on TGX-DDI. It was a really impressive package going over all of the different validation work that had been going on. So we submitted that in 2017. But unfortunately, at that time, the new 21st Century Cures biomarker qualification process was launched. Now the program was revamped, and it's really nicely organized now, and there's very specific set of templates and other things to follow. But what that meant was that after a lot of back and forth and trying to push our qualification package through, the FDA asked that we restart the process basically through their new procedures. So that was tough. We had to basically scrap our PhD thesis and start with developing a new biomarker plan. So we submitted initially to the group. And there was a lot of back and forth on actually finalizing the qualification plan. And we sort of, at that point, had thought that we had finished all the wet lab work that we needed to do. But after many rounds of conversation between the FDA and the TGX-DDI team, it became clear that they really needed a ring trial for us to be fully qualified. And so we redeveloped our qualification plan with the objective of conducting a final ring trial that involved labs that had not done TGX-DDI before. And so one of the main stumbling blocks was the fact that most of the data on TGX-DDI had either come from Georgetown or Health Canada, and they wanted to ensure that labs other than those two labs could actually run TGX-DDI successfully. So our revised qualification plan was submitted in 2021, but the great thing about that was that the FDA was really on board with supporting us to do this ring trial. So they provided us with a grant, a DDT grant, in 2022 to actually conduct the ring trial. The challenge there was the tight timeline. So I can't remember if it was a year or 2 years that we had to complete the full ring trial, but it really put us under a time crunch. But we persevered. That's what we do. The TGX-DDI biomarker working group always perseveres, and we completed the ring trial and submitted it and the full FDA qualification package this year. And that's why we are here to really celebrate. Team history buffs. Did I miss anything? No?
Thank you for that overview, Carol.
Carol, I don't think you missed anything. It was a great recapitulation.
But so Chrissy's going to show us the next slide. What I would say is that, you know, that was a long process, and it sounds like we were just sort of sitting around and waiting a lot. Forget about that. People on the biomarker team did not sit around and wait for anything. We kept pushing this thing forward, and you can see all of the publications that have been coming out of this team over the last 15 years. So beginning with biomarker discovery in 2009 from Hydron and the paper in Nature Reviews Drug Discovery in 2010, right through the validation and further development in 2015s and 16s. That amazing work that we published in PNAS, the external test set validation in 2017. We recognized that bioinformatics could be a challenge for people, so we developed a tool with the National Institutes of Environmental Health Sciences, Jennifer Fostel, to make sure that everybody could do TGX-DDI easily. We recognized the need for transparency and availability of data. So with Andrew Williams, made all that data available in a data in brief. We collaborated with the U.S. Environmental Protection Agency to make sure that you could try different types of analyses. So Chris Corton showed us that you can use this with running Fisher's test and developed a new way to use it and did massive validation work using the correlation engine database. And then we, you know, through this time, evolved from microarrays to qPCR to nanostring to RNA-seq to tempo-seq over up there in the top right with all those different papers. And then we continued to ask, how can we use this in different regulatory applications with those case studies? And finalizing down here, culminating in State of the Science reviews, the first one by Heng Hong in 2020, but really excitingly, the International Workshops on Genotoxicity Testing got on board in 2022. And the outcomes, the consensus opinions of that work group with TGX-DDI really featured there on transcriptomic biomarkers for use in different contexts for regulatory decision-making emerged from that. And these back-to-back publications are all published in Environmental and Molecular Mutagenesis now. So that working group was chaired by Roland. So we didn't sit around twiddling our thumbs waiting for the FDA to approve us. We got all kinds of work done. And over that time, we saw increasing interest from contract research organizations like Innovative, Cytovation, using the biomarker, and really excited to see others using the biomarker and publishing on it. So Ashley got into the game with Stefan over in Procter & Gamble in the top publication, and we have publications from others shown here. So a lot other than the FDA biomarker qualification process has been going on within the working group. I think that's my last slide, Chrissy.
It is. Thank you, Carol. Are there any questions or comments at this point? All right. I think Carol did
Oh,
a really
Chrissy.
Oh, yep.
There's a really nice one from Hydron. I think it merits reading. It does need to be mentioned. It was the perseverance of you, Carol, and Cyril, and the optimism of Yuri, and the continued data analysis by Andrew, and certainly not to forget Hang Hong and Hydron, of course. But I think that's just nice
Oh,
to read out.
It was everybody, you guys. It was an amazing team of really incredible people and new people willing to join in and roll up their sleeves in the end. So it's been an incredible journey. Loved every minute of it. Not that it's over.
It's not over.
We'll talk more.
Most minutes of
I
it,
think
Carol.
I think
Not every minute, maybe.
Carol, to me it's the new people coming in over the years, right? So
Yes.
that we were able to kind of keep it going somehow with new people stepping in and actually making a difference, right? So that's really cool on this one.
Yeah, I agree, Yuri. It's really it's very nice for Hydron to say that, but it's just been such an amazing team effort.
Absolutely.
Yeah, absolutely. Well, thank you both. Speaking of new and amazing people, we are now going to hear from Dr. Vicky Wang, who's at the University of Ottawa. She stepped in, I believe it's been a year now on this project and at Ottawa. And she's just made amazing contributions to both the Ring trial manuscript. She's first author on that. And then she also wrote the draft of the I shouldn't just say draft. She basically wrote the entire FDA biomarker qualification document. So really excited to highlight her and her work today. So Vicky, I know you have your slides, so I will go ahead and stop sharing so you are able to.
Thank you, Chrissy, for the nice introduction. And now I will share my screen. Let me know if you can see my slides. Oops. Guess not this. I'll just share my entire screen and let me know if you can see my slides.
Yep, everything looks good.
Okay, great. So I am Vicky Wang. I'm a new postdoc working with Carol at University of Ottawa, and I'm really happy to be here to share you an update on the TGX-DDI biomarker, and my talk will focus on the results from the Ring trial validation study. But before I get started, I would like to acknowledge the excellent team behind this work. So special thanks for HESI for coordinating this project, and this work was led by the TGX-DDI working group in the eSTAR committee. And we're also grateful for our collaborators from academia, the government, and the industry, as well as the funding support that made this possible. I wonder if I could have my laser pointer. Yes. So now I'm going to give you a bit of the background on the TGX-DDI biomarker. So traditionally, the genotoxicity testing, genotoxicity testing is a key component of chemical safety evaluation. And traditionally, this was done by standard tests, for example, the Ames bacteria test or the in vitro and in vivo genotoxicity assays to measure chromosome damage and mutations. These assays are highly sensitive, but they often give us a false positive results, meaning that they indicate that chemicals can be genotoxic, but after intensive follow-up experiments, they are found to be irrelevant of human cancer development. And these extra testing are usually time and resource consuming. And these assays are not high throughput enough, and they do not tell us much about why and how these chemicals may cause genotoxicity. So there is a clear need for a high throughput genotoxicity assay with improved specificity and also provides mechanistic information and more human relevant data. This is where TGX-DDI comes in. It is an in vitro transcriptomic biomarker that uses gene expression pattern to predicts whether a chemical is a potential DDI, so DNA damage inducing, or non-DDI agent. It was first developed in the human TK6 lymphoblastoid cells. The cells were exposed to 28 prototype chemicals with well-characterized DDI or non-DDI mechanisms. And based on the transcriptional responses, 64 genes shown in the left figure here were identified as being predictive of DDI potential. I just want to highlight the biological relevance of the biomarker here. So the 64 gene panel in TGX-DDI is highly enriched for genes under the regulation of P53, which is the master controller of the DNA damage response pathway. And the idea behind it is very straightforward. If the chemical can activate P53, it's likely triggering DNA damage and therefore potentially genotoxic. This is just a figure from a previous publication by Heng-Hong Lee and her team in 2017. In the study, they tested 45 chemicals in TK6 cells, categorized into 5 different mechanistic classes, but I'm highlighting 3 of them here. Class 1 are just agents that directly cause damage to DNA, so the true DDI, and class 4 are the non-clear the clear non-DDI agents, and the class 5 are the problematic ones that often give us misleading or false positive results in the standard assays. And you can kind of tell on the top panel here, the top 2 rows are the results from the standard chromosome damage or the Ames assays. You can see they did a really good job in classifying the class 1 and class 4 chemicals. So yellow indicates a DDI and blue indicates a non-DDI. They did a really good job in these 2 classes of chemicals, but when it comes to class 5, the problematic ones, the standard assay gave a complete opposite prediction for these chemicals. While the TGX-DDI correctly classified all of the true DDI from class 1 and 9 out of 10 non-DDI chemicals, and they effectively predict most of the class 5 agent. So overall, TGX-DDI has demonstrated high sensitivity and specificity. Going back to this slide, since its first development in 2015, TGX-DDI has been extensively validated in different transcriptomic platforms and cell types, primarily the HEPA-RG cells. And over the years, we can confidently conclude that TGX-DDI had improved performance compared to the standard assays. It's also transferable across cell types and platforms, including the high throughput technologies. And it's also biological informatives, capturing the cellular responses to DNA damage. And it also has a highly objective data analysis pipeline, minimizing subjective or human bias data interpretation. Because we like this biomarker, we wanted formal qualification, like Carol has mentioned. HESI, together with Heng-Hong Lee's team at Georgetown University, initiated this process under the US FDA biomarker qualification program. And we have been through multiple stages over the years. You've learned about the history timeline from Carol's presentation, from submitting a letter of intent to finalizing the context of use statement, qualification plan, and a 2-year of completing the ring trial study. And last year, I joined the working group and helped compile the full qualification package, which is now under review by FDA. So a key part of the qualification process is to finalize a context of use, or to define how exactly this biomarker can be used in a regulatory context. And this is the context of use statement approved by FDA. I'll just read it out. The TGX-DDI should be applied to agents tested positive in vitro assays indicating structural chromosomal damage or clastogenicity, but not numerical changes. So TGX-DDI is not applicable to antigens. And within a weight of evidence framework, it can be used to help interpret situations where the Ames test or in vivo micronucleus test outcomes are negative or inclusive. So in other words, TGX-DDI supports the interpretation of genotoxicity data, particularly in the cases that we have a mixed or unclear outcomes from the standard assays, and TGX-DDI will help clarify whether the positive signal is truly relevant to human risks. And then I will be focusing on the recent completion of the ring trial study, which marks a significant milestone of the qualification process. The main objective of this ring trial was to assess the cross-laboratory reproducibility of TGX-DDI classification outcomes. So 4 independent laboratories participated, and each tested 14 compounds in TK6 human lymphoblast cells at 3 pre-selected concentrations. Everything was done in technical triplicates, and the transcriptomic profiling was done using a high-throughput nanostring encounter platform. And all the 14 chemicals were purchased from a single manufacturer, aliquoted and blinded at Georgetown University. So the researchers didn't know which chemicals that they were testing. And all the labs has followed the standardized SOP for exposure and data analysis. Here's a list of the compounds used in the ring trial. So the 14 test compounds here were carefully chosen from previous validation studies to represent the class 1, 4, and 5 mechanisms. And this sets also include some pro-genotoxicants that require metabolic activations. And 3 negative or positive control were included in each batch of the experiment. 3 concentrations were used, the low, medium, and high, with the highest concentration expected to induce a 50 to 60% cell death. But I will talk about some limitations of using pre-selected concentrations later in my presentation. Yep. So here is a very simplified experimental workflow of the assay. The TK6 cells were exposed to the chemicals for 3 to 4 hours in the presence or absence of the red liver S9 for metabolic activations. And part of the cells were cultured for an additional 20 to 24 hours for cytotoxicity measurement using MTT assays. And we set a cytotoxicity threshold and to ensure the valid TGX-DDI outcomes. So a non-DDI call is only considered valid if the top concentration induces 50 to 60% cell death or when it's over 1 millimolar. The other part of the cells will go straight to RNA extraction, but if S9 was used, the cells were allowed to recover for additional 4 hours before RNA extraction. Transcriptomic profiling was done using this nanostring encounter platform. After quality checks, the data were analyzed using an online tool to classify the compounds as DDI or non-DDI. This was done by a 3-pronged analysis comprising 3 statistical tests, and these methods were used to compare the transcriptional profiles of the test compounds to a set of reference compounds. And then the overall call was done like this. If any of the 3 methods give a positive DDI call, the compound will be classified as DDI, and only when all 3 methods gave negative calls, the compound, and when the cytotoxicity threshold was met, the compound was classified as non-DDI. Because the main objective of the study is to assess the cross-laboratory reproducibility. So we first did a concordance analysis of the TGX-DDI calls across the study site. This is done using 3 metrics, the kappa, adjusted kappa, and AC1, these 3 methods just to assess the agreement levels across the results. And overall, we have a very good agreement in the results, with the coefficients range from 0.6 to above 0.8. And particularly lab 1 and 4, they have a perfect concordance, which is really impressive. And this is showing the TGX-DDI is highly reproducible, even it was done in different laboratories. And the classification outcomes are summarized here. The valid test refers to the number of labs that generated valid results using the assays. The correct calls are when the results are meeting expectations. So you can see the 4 laboratories correctly classify the 3 negative or positive compounds with 100% accuracy. And for the 14 test compounds, TGX-DDI correctly classify all of the 6 DDI agents, again, 100% accuracy. But you can see some tests were excluded for not meeting the cytotoxicity threshold. For example, the stereosporin here, no valid test was generated because the highest concentration was not high enough to induce 50% cell death. So we realized the limitations of using pre-selected concentration. 2 laboratories did additional range finding experiments for 4 of the compounds, and they repeated the assays at optimized concentrations. We're happy to see, so these results are referred as results 2, and we're happy to see that all the repeat experiments generated correct classifications. And remarkably, the accuracy for stereosporin was improved from 0 to 100% when valid concentrations were used. So this is highlighting the importance of concentration selection for this assay. Here just the summary statistics of the same results. If we just look at the optimized data sets, so results 2, the 4 laboratories collectively generated 19 tests on the DDI agents, and they were all correctly classified, achieving 100% sensitivity. And for the 28 tests generated for non-DDI agents, 24 of them were correct, leading to 86% specificity and an overall accuracy of 91%. Just for comparisons, the in vitro micronucleus assays typically has 90% sensitivity and about 65% specificity. So there is a clear improvement of performance by TGX-DDI. In the qualification package, we also prepare a generic test guidance to support the future use of TGX-DDI by new laboratories. So it was adapted based on the SOP used in the ring trial and the practical lessons that we learned during the process. And I'm going to highlight 2 key components in this document, the range finding experiments and the demonstration of proficiency. The test guidance also highlights flexibility in the choice of cell lines and transcriptomic platforms, as well as some additional test considerations before a lab can do TGX-DDI assays. So as I have mentioned, meeting the cytotoxicity threshold is critical for valid TGX-DDI outcomes. So now the test guidance has specified the need for a range finding experiment to select the optimal concentrations to run the test. And this can be done by any assays that can reliably measure cell viability, cell death, or proliferations, or other endpoints. And we also set the cytotoxicity thresholds. So the top concentration has to induce 50 to 60% cell death unless the chemical is already at the solubility limit or the concentration has exceeded 1 millimolar. And we recommend selecting a minimum of 3 concentrations and 5 concentrations as optimal. If a laboratory wants to use TGX-DDI, they are required to demonstrate the proficiency in this assay first. And this can be done by 2 sets of experiments. So the laboratories needs to correctly classify the 3 positive or negative control compounds. And once they have the correct results, they can proceed to to test an additional 5 chemicals, including 2 DDI, 2 non-DDI, and 1 DDI that require metabolic activations. So once they have correct classification for all these compounds, the lab is considered proficient to run the TGX-DDI assays. And to support the future use of TGX-DDI and make this easier, we collaborated with the US Pharmacopoeia to establish the TGX-DDI Proficiency Testing Library. So this is a nonprofit organization. The researchers will be able to buy all these 8 compounds together with quality assurance from US Pharmacopoeia. So just a quick summary of the current status of TGX-DDI and the next steps. So TGX-DDI has been extensively validated in multiple cell types and different transcriptomic platforms. From the ring trial, we know that the TGX-DDI had a high cross-site reproducibility, and it showed excellent sensitivity, improved specificity, and also accuracy compared to the standard assays. And a full qualification package, including a generic test guidance, has been submitted to FDA earlier this year. We're considering to submit a modified version of the guidance to OECD for endorsement. And my co-author Chrissy and I have prepared a manuscript on the ring trial, which is now available on Toxicological Sciences. And in the future, we'll continue to validate the TGX-DDI biomarker in different models. For example, the 3D HEPA-RG spheroids that I will talk about more details about this in my second presentation. We're also interested in comparing and integrating TGX-DDI with other transcriptomic biomarker, for example, GenomeArc. So thank you, but the last I want to thank HESI Global for the stipend that supported my contribution to this project, and thank you for your attention. Now I'm happy to take any questions that you may have.
Thank you, Vicky. What a great overview.
Stop share.
Great.
Have I? Not yet.
Nope,
Okay.
not yet. Yep, we're good. Wow, thank you for that overview. Really nice job. It's a lot of data to get into a few slides. Any questions or feedback from the group for Vicky?
I guess everyone knows this biomarker so well, and we're just really excited about the progress.
Yeah, definitely.
I have a question, maybe just regarding this. Fantastic. Okay, so and great talk, Vicky. But regarding the next steps, right? So because you mentioned at the end, so to develop it is if it's gonna get qualified, right? Then people can start using it.
Mm-hmm.
So and really kind of would be, the next step is using is some really understanding either pharmaceuticals or environmental chemicals, and kind of trying to find a way how that could be the best used, right? So, but then scientifically, are we thinking about developing the TGX-DDI more into predicting carcinogenicity? I mean, like a different context of uses, because this is predicting basically a DNA damage, right? So that's how we have it, and the application in the context of use is to improve a genotoxicity testing. But have you thought about kind of pushing it more, or finding a way how to push it into carcinogenicity risk assessment?
I think that is a really good point. So the context of use that I presented is really for the TGX-DDI qualification, but exactly this biomarker can be used in different applications beyond the context of use there. So for example, as you mentioned, for the assessment of carcinogenic risk. So TGX-DDI can definitely be used as kind of like a first-tier screening of the chemicals, and if we have a positive TGX-DDI call, it may be valuable. It can be prioritized, for example, in risk assessment. Yeah, I guess any suggestions for future directions or next steps, it would be very welcome. Carol?
I love that question. And Connie and Chrissy, you know, we've been really, really impressed with the carcinogenomics working group. And that's all being done on mouse or rat rodent liver. There's fantastic opportunity to go to human liver with TGX-DDI and integrate those, you know, like do some sort of translational study of the biomarkers in carcinogenomics over to, you guys have to finish your mouse and rat stuff first, I know, but you know, in the future to have these translational biomarkers that move from rodents to human cells, and TGX-DDI would fit in very nicely there. So great point by Yuri. We should keep talking to each other and keep this on the radar as future work, I think. What do you guys think?
I completely agree, Carol. I was going to add to mention that work, but also how we set up that other project. And Yuri, I can put the overview paper in the chat, but it's basically looking at things that happen early on, molecular initiating events. I'm sure you're very familiar, but looking at 7 or so that we think are really relevant for rat, that lead to rat tumors mediated through the liver. And so genotoxicity is one, but also some other ones like CAR, PXR. Again, we'd have to think about the translatability for humans and which are relevant, but trying to figure out what are some molecular initiating events that we think really relevant for human, with TGX-DDI being one. I think, you know, I see Patricia's on. We're also thinking later term, some other later term biomarkers with the error-corrected work. So I think it's a really nice way to link a lot of our projects, but we'll take some additional work.
Yeah, pretty cool. Thank you.
Thanks. Hydra?
Yeah, I think so there is a connection certainly, but the TGX-DDI is more on specific mechanism of genotox versus the carcinogenomics is more on the non-genotoxic, which are still difficult to model in vitro. I mean, you probably know all that. You need other cell types, interactions between different cell types. But the more we learn about the molecular initiating events, as pointed out by Connie, the more we can model in vitro, that for sure. So then it could be a model, the TGX-DDI.
Yeah.
Well, I mean, maybe for starters, if you would be able to say, so anything which causes TGX-DDI, you know, DDI response, right? So is genotoxic carcinogen, and because and therefore does not have a threshold or so, and the everything else does have a threshold, right? So yeah, I'm just speculating here, you know.
Yes.
No, just kind of figuring out how one can actually, you know, implement something now.
Yeah, that's right. I think it's about the threshold of the apparently all being genotox, but maybe not being all genotox. And that could become highly relevant depending on what you have in the development or in the pharma development. That's true. So even for the genotox, we may be able to use that more as a threshold mechanism, maybe. Well, we need to get the authorities to think about a threshold in genotox, but the data are there, so yeah, that's right.
Yeah.
Carol, understand. I'm just gonna.
Yeah, I think it was a good question, but I think we have to be careful. I agree with Hadrian about the differences of non-genotoxic and genotoxic mechanisms, and the other part of the eSTAR is more focused on non-genotoxic mechanisms. However, this type of assessment can help us kind of elucidate a little more that overall, the clear classic genotoxicity that we measure. So it could be kind of part of a way of evidence in order to kind of predict, because I want to make it clear, not all genotoxic compounds are carcinogens. They're not all of them. So it would be kind of like if we can generate early data to see if we get there. However, at least in pharma, and I don't know the other companies, but or industries, they if you have a genotoxic compound, you already have the assumption that it's carcinogenic in humans. It's an assumption, even if it's not, if you don't prove it. So, but I think it may have some niche or ideas as we go along. And when we in the GTTC, when we get to understand more about the mutations that we're seeing, mutation patterns, and if we compare that with the GTT-DDI, I'm also thinking out loud. I don't know how, but potentially could work. But it's important just to distinguish the mechanisms because they're completely, yeah. Anyway, that was my thought. Thank you, Gary.
That's a really good point, Patricia, too. This is our systems toxicology committee, but this is the gene tox biomarker. We have a gene tox committee. So how can we link in with the work they're doing and make sure, a lot of the assays they're looking at are complementary, but totally agree with you on, thresholds and understanding and all the nuance. Thanks, Patricia.
Great. Well, thank you all for that input. And I think really nice to hear that we're already starting to think about next steps. We do have a section at the end to continue these discussions. So I'll just move us along if there's no other comments, which I don't see hands up right now. So I'll just go ahead and get us started again. So it's already, yep, we're looking good. It's already been discussed a few times, but just wanted to give like an official update to the FDA biomarker qualification. As Carol gave a nice history lesson on the progress and steps and challenges, many challenges along the way. But really just wanted to kind of orient everyone that, the purpose of the biomarker qualification program is really to develop and qualify these biomarkers as these really reliable tools. And they mentioned that having this specific context of use, which was really nicely presented by Vicky, that we can provide evidence that the biomarker can be reliably used in multiple drug development programs for things like safety monitoring. But I do think thinking about how we could maybe adapt our context of use moving forward is a really nice idea. So with that, in 2025, we submitted the full biomarker qualification package with the ring trial results in March. And we received a response so FDA did an initial review and had some response for us. We were able to go back and give them some more details that they asked for in July. And then in September, we received a notice that the package was marked reviewable, which is very good news. And we were asked for some clarification to outstanding questions, which we responded as well. So with them marking our package reviewable, they have about 10 months to review the package and give us a response. So hoping to give you all an update within, hopefully early 2026, but we'll see how long things take and we'll keep the group posted. So far we've heard from the history of where we were. We've heard kind of where we are now with the package submitted, and now we're starting to think about where we're headed. So I'm going to hand it over to Dr. Andrew Williams from Health Canada to give us a presentation on some of his work on the updated PCA analysis that he's been working on for the tool. But I also just wanted to give Andrew a really big thank you for all of the work he's done on the ring trial, and he was our statistical mind behind a lot of this. So we appreciate his work and continued work, which we'll hear about right now. Andrew, do you want to control your slides?
Yeah, I'll try.
Okay.
There we go. Let's see. Okay. Are we? It's not in presentation mode, eh?
Yeah, not in presentation mode.
Here we go.
Yep, looks great.
Sweet. Thanks for the introduction. A lot of the work I'm presenting today is kind of something I've been thinking about for a while. It kind of a discussion with Julie Buick at the Health Canada Science Forum when I was looking at her poster presenting. I see Tara's on, so presenting some of Tara's data. So this is going back to February. So this presentation is looking at objective inconclusive zone to enhance the PCA-based classification for the phylo biomarker. I'm not going to spend too much time on this slide because everyone here knows what TGX-DDI, the biomarker, is. 64 genes. We implement a three-prong approach. So a probability analysis based on the shrunken centroids, hierarchical cluster analysis, and a principal component analysis. So I'm just going to briefly go through the three-prong approach. And I'm going to use the data from the ring trial. So another plug on the ring trial in the bottom corner there. So the nearest shrunken centroid classification is a method that classifies a new test agent based on their gene expression profile relative to the reference compounds or the reference centroids. We use the probability of class membership, which is an analog to the Gaussian linear discriminant. And probabilities are given values between 0 and 1, and we define probabilities between 0.1 and 0.9 as inconclusive calls. So in this, I'm going to say in the hierarchical cluster analysis, you can see we cut the tree here where we have the DDI compounds on the top and the non-DDI compounds on the bottom. And here you can see that the low dose for chemical A, which as Vicky showed in her slides is a toposide, is with the non-DDI agents or reference compounds. And when we look at the mid dose, we see that it's now clustering with the DDI, and at the high dose with the DDI. So here we would give it a DDI classification based on the fact that it's clustering with the DDI agent. So the idea here is that, you know, what is this unknown test agent closest to? So it's closest to the, the DDI reference compounds. We can also get inconclusive calls in the hierarchical clustering analysis, and that's basically when the cluster analysis, the new test agent clusters as a singleton, or there are situations where you can get this phenomenon called chaining, which actually it's in a situation where it really disrupts the cluster, the clustering of the two clusters. So it's in a situation where the new test compound chains in a very unexpected way. So that there we would also give an inconclusive call. But for the PCA analysis, we don't really have an inconclusive zone. So we look at this test compound, we can see that the low dose is in with the non-DDI agents, the medium and high dose are with the DDI agents. And when we initially did this, you can see that, you know, the PC1, first principal component, is really a contrast between DDI agents and non-DDI agents. So why establish an inconclusive zone for the PCA classification? So what we want to do is improve reliability, consistency, interpretability of the TGX-DDI outcomes, in particular with some of the other, let's say, classification methods. You know, like I said, it was a contrast between the DDI and non-DDI agents. This threshold of 0 was arbitrarily chosen. But we can see that there's let's say, a uncertain zone between anything just to the right of sodium arsenite and say to the left of course, the name's failing me right now, but the 2%
ethanol.
Ethanol, thank you. You can see between these two, there is, you know, let's say an area of where there's no density with regards to the reference compounds. So we can say that there's somewhere in between these two blue bars or horizontal vertical lines, is that there is some uncertainty with regards to the classification. So that, for example, if a new test compound fell on the red line, what would you give it? Would it be DDI or non-DDI? And this is, we've seen some data like this in the past. So to give it a clear DDI, non-DDI call with chemicals falling within this zone, there's a high level of uncertainty with regards to that call. So again, it's false confidence of calls, inconsistent interpretations across analyses. So just going back to the probability analysis. So this is what the discriminant scores look like. Here's how the class probabilities are calculated. In the graph to the right, I've put in what the DDI centroid and the non-DDI centroid looks like. So can we use, how we define the inconclusive zone in the probability analysis and the PC analysis? I just want to throw something in here, the bisection method, which is a root finding algorithm. There's other ways to do this. You could brute force this, but in terms of an elegant, simple algorithm, you start with two starting points, A and B. You evaluate the function at both of these points. And what you want is the sign of f of A and the sign at f of B. They have to be opposite so that you know that the function crosses 0 somewhere in between those intervals. And there's an assumption here that it only crosses once, but when we're looking at the probability analysis, that function is a monotone function, so that's easy. We know it's going to cross once. So you take the half interval C, and depending on the sign of f of C, you cut the interval in half. So now you look at, here, then it cuts down to here, and then eventually you just keep shortening this interval until you get to the root. Okay, so this is just a simple root finding algorithm. So so how do we do this in the probability analysis framework? So we have the probability pk of x minus some threshold that we want. So here again, 0.9, 0.1. Now what we want to do is we want to project the data from the 64-dimensional space into the 1 or 2-dimensional space. So we're projecting the data based on the principal component analysis. And we want to use this to calculate, what the thresholds for this uncertainty zone should be. So if we do this, then we can see that there's so the line, the dashed line to the left is what if a probability of 0.9 would be, and the dashed line to the right is the value at 0.1. And one thing to kind of observe here is that both these lines are actually left of 0, which is kind of interesting. So, this would define an objective inconclusive zone for the lower and upper thresholds. And that, like I said, both of these values are actually less than 0. So definitive DDI calls are now reserved for samples with a PC1 that falls below the lower to the left of the leftmost threshold there. So this approach reduces the risk of misclassifications near the decision boundary and improves consistency with the nearest shrunken centroid analysis. or anything. So conclusions and future directions. Inconclusive PCA region improves the interpretability and transparency. High confidence calls are reserved for extreme PC1 values, minimizing discrepancies between analyses. Probability-based thresholds provide an objective, systematic approach for the biomarker classification. Again, it's one of these things where, you're looking at things on the line. It's like, are we comfortable, with the current approach or not? And, this is something I've been thinking about for a while, and chatting with Julie at the Health Canada Science Forum is like, and also this has kind of popped in some work with Paul White as well with his Tox 21. So anyway, this is some work that I'm currently drafting up as a EMM brief communication. It's still kind of ongoing. It's just like everything else. There's a fair bit of things on the go, and it's slowly moving forward. And the opportunity, thanks Chrissy for me presenting this results, kind of helps with that. So we do have some times for questions or clarifications. I'll stop. Actually, I'll keep sharing just in case someone wants to go back on a slide.
Yeah, thanks,
Andrew, this is Yuri, and Andrew, this is Yuri.
Andrew.
Just sorry, jumping in. It looks pretty cool, but my question would be: okay, there is always uncertainty in any test or any analysis, right? So if you look at current classification of DDI and non-DDI, and if you implement this, what problem does it solve? How this implementing the uncertainty kind of in PCA would help? I didn't get it. How that would help the current interpretation of the data?
Yeah. So
Does that make
right
sense?
now,
What I'm saying?
yeah. So right now, we need at least one of these to be DDI.
Yep.
And so say the probability analysis, say it gives an uncertain classification. And then, for example, you look at the hierarchical cluster analysis, and it clusters as a singleton. So it's nowhere. So in terms of, it's in a space of low density relative to the reference samples. Then you go to the PCA, and it sits on the line. And if it sits on the line just to the left of it, it's given a DDI call. So the question is how confident are we in calling that a DDI agent if it's generated by the PCA solely? And you visually see it and you're like going, do I believe it?
Yeah.
It's that situation. It sounds extreme, but looking at some of the data poor chemicals, these types of things pop up. And if you're running at high throughput, right, when you don't say you're not looking at the pictures, right, you're just generating DDI call or a non-DDI call or an inconclusive call. If you don't look at it, then you may not question it.
So if it would fall into this inconclusive, you would just basically say it's all inconclusive, you don't know. And you would be confident about it instead of calling it DDI and just saying, man, it's just kind of like right here. And it could have been here on the other side too, right? And then
exactly.
It has the same weight in terms of kind of an understanding of the risk because it's DDI positive, right? So that's this is actually really cool, and I understand that it would be very helpful for especially the high throughput kind of applications, because then you would have a better kind of a granularity for the data. Thank you so much.
No, great question.
Yeah, thanks. That was really helpful. Any other questions? Oh, I see Logan in the chat asked if you recompute the PCA each time you have a new compound, or whether the vectors are fixed based on the reference compounds.
The vectors are fixed based on the reference compounds. Yeah, so it is fixed so that yeah, there's no influence of the new test compound.
Any other Yep, Carol?
Yeah, thank you, Andrew. I think this is, as Yuri always says, it's cool. So I'm going to say it's very cool. I think it's really important. Can you go back one slide? So I know I remember the conversations as well with Julie and Paul when we were seeing weird things with the data poor chemicals. This is going to be really helpful to help clean up some of those data sets. And you're drafting an EMM brief communication. Do you need more data to test, or do you have enough? You know, are you looking for anything in particular from the group in terms of input or help? And I'd also like to ask the group, you know, because at some point, do we change the classification criteria to implement this? So it's where, you know, we don't want you to run off and publish a paper that says you should do this if the group doesn't say this is what we should do moving forward with TGX-DDI. So I think we need sort of buy-in from the group, and I'm wondering what you need.
Yeah, in terms of, you know, there's like I ran this all on the ring trial data, and there's no real examples of edge cases.
Mm-hmm.
You know, I definitely know there is some in Tara's data. I know some in Paul's data. I know we have some inconclusive calls based on the PA analysis in previous data. I mean, I think that, you know, there's a couple different ways you could look at this. You could start collecting, you know, actual data that was inconclusive based on the PA call, and then see, you know, where they fall in the PCA analysis.
Mm-hmm.
But no, I'm very open. So, I mean, if there's thoughts, if there's data, I think there's other ways of tackling this problem as well. But I think the idea of having some type of fuzzy classification, it's like I said earlier, it's something I've been thinking about for a long time. And then when you see the examples, and it's like, I have to go back to see if Anne-Marie's data, if we had some of those edge cases as well.
Mm-hmm. But I guess it would be useful. So, you know, what we'd want to do is update the proficiency, or not the generic test guideline to implement this. But we can only do that if the group reviews the paper and decides that they want to do that. So,
well, yeah, and there's other steps too, right? Because we have the online tool, you know, it's, you know, talking to Marcus to say, oh, you know what, we would like to make a modification. So no, it's just not, you know, I definitely agree that this has to be a group's input. I mean, it's just you know, how my, you know, just me and my thought processes, and I'm looking at this and I'm like going, oh, I don't like this. We can do better, or I feel like we can. But yeah, I mean, you know,
maybe that's something that Chrissy could set up. Then we could have a more extended conversation about it, and Andrew could share what he's done so far with the paper and other things. Yeah, Chrissy and Connie.
Yeah, absolutely. I think that's a good next step, and we can kind of think through the timeline and process of having the group review it, and then if we decided to adopt, what are the multiple checklist items that we would need to complete if we wanted to move forward. Yeah.
Great. Thank you. Thanks, Andrew.
Thanks, Carol. Yuri?
Yeah, I just want to say a similar thing with what Carol said. Maybe kind of really think about it, and for Andrew, whether this group can help, right? Because I think this is we might consider, I mean, think about this as a kind of a future development, because I think this is important, right? So, and as a future development work would be fantastic. And if we can help somehow, or the people in the group can help to this and develop something around it to facilitate your work, Andrew, that would be, I think cool stuff to think about. That's it.
Thanks. Yeah, I agree. Happy to take this further, set up a meeting with our group and whoever's interested, and yeah, we can continue these discussions. But thanks, Andrew, for your presentation. Really interesting work.
Oh, sweet.
I say one thing, Chrissy, I totally agree, Andrew. Thank you very much. But we worked for the ring trial team for a long time, but this might be a time to kind of re-gauge with eSTAR generally and see who wants to come to these, if we do monthly meetings or project meetings. So that might be something for the team to think about too.
Yeah, thanks, Connie. I know we did have people who were interested over the past year or so, but it was hard to get them engaged with the ongoing ring trial. So completely agree. We can send a note around to all of eSTAR. Awesome. Okay, great. So I think we'll just continue these really nice thoughts from you all and looking to the future of the project and the committee here. So one thought that has come up a few times is to kind of create this nice full circle moment. Carol had pointed out that this had been TGX-DDI had been kicked up on a Nature Drug Discovery article. So, maybe it would be nice once this biomarker reaches qualification that we could have a opinion piece or some sort of article that describes the process and highlights all of the really nice work. So I think this is a great next step. Would be happy to engage with whoever wants to be contributing to write this, but I think that's something we'll put on hold until we finalize the qualification. Any thoughts on that before I move us on? Okay. So I know Vicky has been working on some extended TGX-DDI experimental work, so I wanted to give her some time to go over that now and talk, get some feedback from the group about future steps. So Vicky, I'll hand it back to you.
Yes, sure. Can you see my slides?
Yeah, looks great.
Great. Hello, okay. Hello, it's me again. So now I'm just going to talk about some ongoing work that I'm currently working on, which is focusing on validating the TGX-DDI biomarker in the HEPA-RG 3D sphereize. So this is being done in collaboration with Dr. Lauren Carter at Health Canada, who has a lot of experience with culturing the primary mouse hepatocyte sphereize. And we're now working on optimizing the protocol for the HEPA-RG cells. They have also established a HEPA-RG cell working bank in her lab. There are a few things that I wanted to do in this project. There are always a lot of things that we want to do, but first, I want to establish and optimize this HEPA-RG spheroid model for TGX-DDI testing, and eventually to validate the biomarker's performance in this model. And I wanted to compare the transcriptomic profiles between the 2D and 3D HEPA-RG cells. So we do have data from 2D HEPA-RG cells in the TGX-DDI validation studies, but it will be interesting to see if the higher physiological relevance can improve the TGX-DDI predictivity, for example. And I'll also categorize the key pathways or the cellular responses induced by the genotoxicants using the TEMPO-C code transcriptome assay. So this is something kind of aligned with what we talk about, the potential molecular initiating events, and maybe like setting up like a data set to set up a threshold. Definitely something that we can explore using the data set. And since I will have the transcriptomic data sets anyways, it will be interesting to evaluate and compare additional biomarkers, for example, the genome mark in this 3D model. So after rounds of discussions, we have finalized the experimental designs. So I have two designs here, both employed and repeated daily exposure for 3 days, but the difference is the time point. Let me get my laser pointer. Yeah, but the difference is the collection time point. So first at 4 hours and the other at 24 hours after the last exposure. So the first design worked well with the 2D HEPA-RG in a previous paper, and it's a design used by a previous validation study, but I believe that was done in TK6 cells, and it's also a collection time point consistent with our ring trial study. And design 2 is sample 24 hours after the last exposure. It aligns with the genome-mark design, and it also worked well for the TGX-DDI in 2D HEPA-RG cells in the same paper. And it was a design used in the NIHS chemical screening paper. So both of these designs are kind of already being used by previous studies. We wanted to capture both early and more sustained responses and to kind of see if that worked for TGX-DDI. So here's just a very simplified experimental workflow. On day 0, the frozen differentiated HEPA-RG cells will be thawed and then plated in a specialized 96 well plates at 2,000 cells per well. And on the next 10 days, these cells will just self-assemble into stable spheroids. Of course, I will change the media in between. And then on day 10, the spheroids will be exposed to the TGX-DDI proficiency test compounds at concentrations defined by preliminary range finding experiments, followed by 3 days of repeated daily exposures. So on day 12, it will be the last exposure, and the first collection will happen, the collection will happen 4 hours after the last exposure for design 1. The spheroid lysate will be collected for the whole transcriptome tempo-seq analysis. And then on day 13, which is, it will be the 24 hours after the last exposure, the spheroid lysate will be collected, and here I'll also collect the conditioned media for parallel cell viability measurements. So just a quick reminder of the list of the proficiency test compounds. This includes the 3 negative or positive control from the ring trial, and then 5 additional compounds that are DDI or non-DDI with or without metabolic activation. But the good thing with HEPA-RG cells is it has metabolic competence, so we don't need to use extra like the exogenous S9 system for these chemicals required additional activations. And the US Pharmacopoeia kindly provided part of the test compounds for this project. So I'm currently in the stage of testing different cell viability assays and looking for an optimal concentrations to be used in the test. So the first 2 assays can be done using the conditioned media, including the LDH release into the conditioned media and the measurement of albumin concentrations using an ELISA kit. And the last 2 assays are will consume the spheroids, including a life and death staining, followed by a confocal imaging, and also a kit that measure the ATP contents in the spheroids. But ideally, I want to rely on the assays that use the conditioned media because I can still use the spheroids for tempo-seq assays. So here are just some fluorescent images of the spheroids that I've got after 72-hour daily exposure. So red indicates the viable cells, and green indicates the viable cells, and the red indicates the dead cells. I won't go into details here because these are really preliminary, but I want to highlight a few key observations. So the first, I noticed that the spheroids that I have variable size and shape. I'm now optimizing the culturing protocol, and it seems that the new procedure is now giving me a more consistent and more rounded spheroids. I'm hoping that will kind of improve the reproducibility of my results. But with that said, it's not yet clear if like this varying sizes or shape will affect responses of the spheroids to the chemicals. And the second thing is there is a clear visible cell death at higher concentrations, showing that this assay is able to capture some dose-dependent cytotoxicity. But again, this is a quick screening assay, and we still need to develop reliable methods for image quantification. So I will end with a quick summary of my current findings. So I have done 3 of the assays, like both are preliminary data. But for the LDH assay, unfortunately, it's just not sensitive enough for the HEPA-RG spheroids, at least with our experimental design. And for the human albumin ELISA, the preliminary data have high variability across samples, and it's not yet clear if it's from the assays or from the model itself. And I've been reading some literature, and it seems that we may need to normalize the results to the total protein contents, because this will definitely be affected by the number of cells in the spheroids. And again, for the life and death staining, as I have shown you before, it works qualitatively, but we need to develop methods for quantitative image analysis, and I believe Lauren is currently working on that. And I'm hoping that I could kind of like quantify the images that I have so far. And now I am testing the last assay. It's a kit that measures the ATP using the spheroids. It's actually the most commonly used assays in previous publication for 3D HEPA-RG cells. And so far, and it appears to be the most promising assay for quantitative cell viability measurements. Okay, thank you for your attention. That's just a really quick update of the project.
Thanks, Vicki. Really exciting, and I know it's great to see those images. It's a long process to get there. Any feedback from the group on these initial kind of first steps by Vicki and where it's headed?
Just think it's really exciting. I know a lot of people are using HEPA-RGs more. Is this more human relevant? And then the 3D, if there's a course of interest. So I think it's really important to see how the protocol performs, what needs to be done, but it looks really promising. Thanks, Vicki. I did put it in the chat, and maybe more to Steve a little bit, or I'm sorry, to Carol, but just a note that you probably know, and I know you work with Steve Ferguson, but he's in this space, so it might just be interesting to pull him into some of these conversations when he's back from the shutdown.
Yeah, and there's also Carol Guo, who's done a lot of work with the 3D HEPA-RG hi-fi PacBio sequencing and duplex sequencing. So we've been making connections with others who are working in 3D, and talking to Ashley as well, who's doing that. So, but yeah, Steve's an obvious person to go to as well.
Yeah. And
Thanks,
I think maybe a question for Vicki and Carol, is there anything that our team can do to support? Would it, as you move forward, if it's having meetings to get feedback, or are there connections that you need? I mean, you are very well connected, but anything that you can think of that we can help with, I'm happy to do so.
Yeah, thank you, Chrissy. I mean, we're talking a lot with Ashley, but when Vicki's got her protocol locked down and looking good, I think it would be great to connect with the whole team.
Okay. Yeah, it's really exciting. I know we touched base about the cell painting work that Oasis is working on, and we're interested in the 3D models as well. And I know, Carol, you mentioned maybe that would be a future direction, so we could always combine on that when you get there and get things up and running.
Yeah, eSTAR and GTTC, there's a lot of enmeshed and overlapping projects that are really exciting.
Yeah. Hydron.
Yeah, where did you, so your HEPA-RG protocol, I didn't get it completely. Do you spin when you set up the spheroids, or?
Yes, you after seeing the cells, you just spin the plate for 3 minutes,
Yeah,
uh,
okay.
kind of
Okay.
to ensure all the cells are at the bottom of the plate.
Yeah.
And it was recommended by the company like Biopredic.
Yeah. Yeah.
We first kind of leave the culture plate on the like a slanted with like a tilted plate, and then it's kind of have an angle to kind of facilitate the cells to aggregate for the first a few days. So like the first, I was mentioning that I'm kind of optimizing the protocol to have like a more rounded spheroids. So what we have been doing originally is we just leave the plate with an angle over the 10 days, and on the date of exposure we'll just lay it flat. But after we have been talking to different groups working with the
Okay.
3D cells, and it seems that if you leave the plate flat during the self-assemble phase, they can kind of aggregate more naturally instead of kind of putting a force or pressure on them. They
Yeah.
tend to form a more rounded spheroid, a more consistent spheroid.
That-
So I'm
that-
now trying the new
Yeah.
protocol.
Yeah, that's so we usually do it with human primary hepatocytes, and they form nice round spheroids. We don't have experience with hepatology. It's just a protocol from tissues where they recommended a spin, but we never did it actually. They say very shortly 300 G. So for the primary human hepatocytes, we never spin. We just let them sit,
Mm-hmm.
and then they seem to like that.
Yeah,
Okay.
that's interesting. That's definitely something that we can try. And in one of the discussion that we had, the other group from French, they also they didn't spin down the spheroids because they think that would kind of too harsh to the cells.
Yeah.
But I think because Lauren has been working on spheroids for quite a long time, and
Yeah.
it seems that like spinning the spheroids didn't really affect the cell viability, etc. Yeah, but it seems to be working. And we did like a 500 times G for 2 minutes.
Yeah, maybe you do it because the tissues protocol says 300 G. Maybe you try it a little bit lower. I don't
Yeah,
know.
yeah,
I mean, I think you have to try it in your hands, and it has
yeah,
to work for you. So maybe something around that trying out could help.
yeah. That's
Yeah.
a really good suggestion. But I guess there's one last thing that I wanted to mention, that instead of we're using a specialized plates from Inspiro. So
Yeah.
we're not using the U bottom
Yeah. Yeah.
ultra
Yeah. Yeah.
attachment. So like the specialized plate, it has a like a kind of have
Conical.
a
I know we use that too partly. Yeah.
Yeah, so I'm thinking that what Lauren and her biologist has noticed, like sometimes the cells tend to sit if you don't spin down. The cells tend to sit on the edge of the
Side.
plate.
Yeah. Yeah. Yeah.
So kind of the
Mhm.
spinning may be necessary, but we'll try to maybe lower the speed. Yeah, just to
Yeah.
make sure the cells are at the
Yeah.
bottom. Yeah.
Okay.
Great. All right. Well, very exciting, and we'll be looking forward to hearing updates as you move forward. We will go ahead and pass to Carol to talk about some ideas for OECD.
You know, the ironic thing, gang, was that we decided that we would go to the FDA because it was going to be too long a process to go to the OECD. And I'm not so sure about that now. But it does make sense now to go to the OECD. And the timing is right for a few reasons. First, I'm showing you the OECD test guideline for guard skin. So there's now an endorsed test guideline for a transcriptomic biomarker. And when you look at the history of guard skin and even how guard skin is used and applied, their guidance, it's really, really similar to TGX-DDI. The other interesting thing that's going on is that the OECD is actually now asking us to bring this biomarker to them. They don't want, and it doesn't make sense to have individual biomarkers go through test guidelines though. we're going to have a lot of biomarkers. To have a test guideline for every single transcriptomic biomarker doesn't make a lot of sense. So their idea is for us to work with, and actually, let's go to the next slide first, Chrissy. Just in terms of how big a lift this would be, I'll remind you that we now have this generic test guidance. And guess what was used to create the generic test guidance? To create the to follow a template, we used GuardSkin. And by we, I mean Chrissy and Vicky. Thank you guys. So we're not all that far away from developing a test guideline to begin with. And we borrow the OECD language in this test guideline, in this generic test guidance. We have the proficiency test compounds as we've described, and their availability from the nice work that Brett Howard's done for us at USP. So I feel like we're pretty close anyway. The other thing to bear in mind is that usually the first step for OECD launching some sort of test guideline development is a detailed review paper. And just this year, the International Workshops on Genotoxicity Testing conducted a meta-analysis of all the transcriptomic biomarkers for genotoxicity. And so that work has pretty much been done for us. So timing is right. Now, OECD has come to us and they've said, we don't want, sort of standalone. We want transcriptomic biomarkers to go through the test guideline program, but we don't want standalone biomarkers. And so Birgit Mertens, who leads the work with GenoMark, is very interested in potentially leading or co-leading a team to bring those two biomarkers together. We have published papers showing how nicely they integrate with each other, that they give complementary results. And even if you do benchmark dose modeling on the genes within those two biomarkers, they give identical points of departure. So GenoMark was developed for HEPA-RG, TGX-DDI for TK6. So the proposal would be to move forward with those two as a pairing and see what the OECD thinks of that. Another opportunity would be to bring in some in vivo biomarkers in parallel, in vivo transcriptomic biomarkers, but we're sort of, Birgit and I were sort of thinking about maybe just taking in vitro together on its own. So I'd like to hear from the group as to, is there interest in this? Would there be volunteers? I don't actually know, and I'm not sure if Cyril's still on here, how, and I see Tara's on, so to call Tara out, how HESI interacts with OECD and how that works, because I know you need lead countries to take charge of the process, and so I'm not entirely sure how this would work. So I'd love to hear people's thoughts on this.
Carol, can I just ask, is the thought it would still be a test guideline, but for both biomarkers, or it would be an IATA?
It would be a test guideline. So we'd have to develop a proposal, first of all, together. The first step would be to develop a proposal and submit it as to what exactly we want to do. But I think they want a test guideline.
Thanks.
Yuri?
I need to get off the mute. So my question is, like, test guideline for transcriptomics biomarkers, having one for all of them versus having a test guideline for each biomarker. I don't think you can have a test guideline for all of them together.
No. You'd bring them in batches, you'd bring them in sets that are meant to be used together or that follow the same. So that's why GenoMark and TGX-DDI would go together, because they're both for evaluating whether chemicals are genotoxic or not.
And the point would be that you could use either, or how would that
we'd have to work that out. You could use either. I mean, if you're running an experiment and you have a transcript and you've developed you've done RNA sequencing or something, you can readily apply both those biomarkers at the same time.
Yeah, no, absolutely, right. But it's all about, like, that would need to be specific for each essay regarding like cytotoxicity, how you run that, right? So it's not going to be kind of a one guideline which would cover both. It will be so they just want to have a kind of a lump them together for the process that they don't need to, because they are very similar, right?
Yeah, if you look at the test guideline for skin sensitization,
yeah.
So the drug, what's it called now? Guard skin is part of a set of NAMs that are used to together in an integrated test to determine if something is a skin sensitizer or not. So it's the same kind of idea. You'd put them together. They'd probably be little individual mini guidances within a larger guidance. If you know.
Okay.
Yeah, that's what they did. That's what they did for skin sensitization.
Yeah, but that's right. But this goes after these, the variety of pathways, right? Both of these biomarkers it's basically one pathway. It's all P53 what we are doing, right? And
different sets of genes. Yeah,
just different set of gene, but but the outcome is it's basically the same, right?
Yep.
So the question is, would that be then required to? I mean, but I'm sorry, I'm gonna stop because I'm just kind of thinking loud here, and that's a discussion, like a separate discussion now.
Yeah, I mean, there'd be a lot
to be applied,
lot to discuss. Yeah, how that would look.
We need to talk about it. Put it in that way.
Yeah.
And I stop.
Yeah.
Maybe I could just add to Carol and Yuri for guard skin. And Carol, you said this, but they again linked it through the AOP pathway, and that's how the skin sensitizations like framework are. If you determine something is or isn't, they target different key events. So maybe that's a way we propose it. Still, if we're talking about carcinogenesis or something, but they measure similar pathways.
The problem is that TGX-DDI doesn't target a key event or a molecular initiating event. It's a bucket. It captures all the different molecular initiating events that lead to genotoxicity, and then genotoxicity isn't a key event. It's
exactly.
Yeah.
It's different. It's different, and it has a our context of use is actually in my mind very useful and practical. But then change it in. We just need to talk.
Yeah. I mean,
it's a great discussion.
The big question is it worth the effort to bring this through the OECD? And do people want to do it? And are there any volunteers to be involved? Those are the big questions.
I think the first question would be, what is the advantage to do that? What would be the impact to bring it through the OECD?
Advantage.
Right.
Pardon, Connie?
I think it would be a huge advantage to have a test guideline. I mean, that's kind of, I know we're doing the FDA qualification, but to me, that's what's still seen as big V validation.
Mm-hmm. I don't think people are going to use TGX-DDI unless there's an OECD test guideline.
And I would agree with you, Carol. So I think we should talk.
What do others think? I mean, this would be a big job to take on. Everybody's lifting up their hands to get excited and want to get involved. I think people are afraid if they say anything that they're going to be recruited to write an OECD test guideline.
I mean, maybe I can just ask from the group, and feel free to use reactions here, like maybe a thumbs up if this is something we just want to start exploring a little bit more. So, setting up those initial calls.
It doesn't have to be done within HESI, right? Like, so if HESI's not interested, then that's fine. But yeah, we need to know.
I mean, I think HESI would be interested, but we need a group that's willing to. Mm-hmm. Oh, John, nice to see you.
Hi, can you hear me? I just got a message. Yeah. Okay, cool. I think that this is by far the most developed transcriptomic biomarker for anything that we have. Mm-hmm. And like, if you're going to pave the way for the use of more transcriptomic biomarkers, this is the one to do it with, right? So I say do it.
You say do it. I love that. Pave the way. I remember a couple of years ago, I had pictures of pioneers on one of the slides when I was presenting.
Thanks, John.
Yeah, notice I didn't volunteer.
I noticed that too, John. Yeah. Thank you. All right. Well, I did see some thumbs up and other things. We could maybe do a little poll or something at some point. Oh, Tara, there we go.
Yeah. I just wanted to weigh in. I think definitely it's worth initiating the discussion. I'm not directly involved in WNT, but quite heavily involved in WPHA as chair for the last number of years, and these are certainly the advances that we're looking for. And I think certainly to bring it to the OECD at the level of discussion, and maybe talking a little bit about what is the process and what is the pathway to bring this through to a test guideline might be an interesting first conversation. I do know that, with the updates of some of the guidance for validation and advances in the context of the IATAs, there is quite a bit of discussion around, how do we bring these through in a process that helps us, I don't want to say expedite, but maybe find efficiencies in getting to that tech guideline. And maybe this again is one of those examples where you've gone through such extensive steps already in the ring trials. And, I think I support what John is saying in terms of the level of readiness of this particular biomarker. So definitely I would also support at least having those initial conversations with the OECD to see, if this is a example of something that we could bring through that process in an efficient way.
And Tara, do you think that the like through Natalie Delrue and EFSA would be the right path to go? I mean, the EFSA stands for Emerging Science. No, what is it? ESCA, Emerging Sciences and Chemicals Assessment Advisory Group at OECD.
Yeah, certainly that would be, I think, a nice place to start, because ESCA really serves as that intermediary body, if you will, between the WPHA and the WNT. So it could definitely be a nice starting point.
Okay. So maybe we could do a little survey, Chrissy, of who would like to be involved in that. And I mean, I don't know how it works in terms of setting up. I've never developed a test guideline, so I don't know how it works in terms of setting up a working group or anything for that.
Yeah, I'd be happy to start looking into this, and Carol, we can connect about the first people to talk with. But I completely agree. I don't see why we wouldn't at least have the initial discussions and see who's interested. It seems like a pretty good place to start, and then we can make some big decisions later if needed.
Perfect. And we could also talk to George Douglas. And he knows the OECD inside out and backwards, and is now working with Stephanie Smith-Rowe and Anthony Lynch and Talita on the ECS project.
Okay, great.
And in Canada, it would be Tim Singer still, I think, Carol.
Okay.
Thank you.
Thanks, Tara.
Okay, thanks.
All right.
Great presentation. Thanks for all this fabulous work.
Wonderful. Well, thanks to hear from some of our members. Really nice to get your input. We have one last piece here, just an outreach strategy that Connie was going to go over. So Connie, I'll pass to you.
Yeah, I think this is really in line with what we just talked about and all the work we've done. It's really to propagate and get it into other people's hand, right? We want other people to use this biomarker for it to be useful to help make decisions about chemicals, be it either in the screening context or for that formal, submission package as part of a weight of evidence. So one of the things we were thinking too is that, in the ring trial, we had these labs go through, and we have a really nice SOP that Vicky flashed and we can show. We're talking about the validation in those two different contexts. But one thing to think about is, not everyone's going to run it in their own lab. So could we have contract research labs potentially do this? And I know at one point it was offered by one, but I believe they no longer have wet work at their site. So I'm not sure right now if there is anyone who could do it. I don't think Florence is on today. She said they're going to offer it. Florence Bertelson was part of the ring trial. So they said, I think later this year, they're going to put on their website. But just kind of wanted to put out to you all thoughts about how do we, again, kind of propagate this. If you're at a CRO, I'm looking at names, I'm not sure if anyone is, what would you need to maybe take it on? Or if you have ideas for people we could reach out to. I reached out to the Institute of In Vitro Sciences, IIVS. We work with them on some of our other projects. I didn't hear back yet, but just kind of to gauge interest. But other ones, if like Charles River, Innovative, if there's either contacts or thoughts about how we reach out to them or who we reach out to. But again, just to make these a little bit more, turnkey, people could order it if they wanted the study done.
Thanks, Connie. Yeah, if there's any thoughts now, we're happy to hear them. I'll just put up the last slide here, which has Connie and my email if you don't have it, but happy to hear, more thoughts, or if you have contacts, that would be really helpful for us.
The other thing I'll say too, I think if people in their own lab are excited about running this and trying this, you can reach out to us. I think we have a nice group that have a lot of expertise. So if you have a protocol question or a thought, or just to let us know that you're doing it, I think that's really exciting too.
Oh, I think, Ulrika, you're on mute.
Yes, true. So I can do some advertising and contact our usual CROs to see whether there's some interest.
Oh, excellent. That's wonderful.
Yeah, and we'll share that around when I see it online. She said, I think Q4, which is now, but I'll keep an eye out for that and can share it.
Mm-hmm. Great. Okay, thanks, Connie. All really exciting updates, and thank you to everyone who's been on for our full 2 hours. We have a few minutes left, so I just wanted to make sure I got everyone's input. So if you have any burning last questions or thoughts before we close, please go ahead and raise your hand or drop it in the chat. I'll give people a few seconds.
I want to say thanks to Chrissy and Connie for all the incredible leadership too, and helping keep us moving forward one step at a time. Thank you, you guys. We wouldn't be here without you for sure.
Thanks, Carol. Thanks all. We appreciate that. But I will definitely echo what you said earlier. It's been a wonderful team to work with. I feel very honored to work with people who've been on the same project since I was in high school, I think. Yeah, it's been a wonderful team, and yeah, you all have done a lot of work, so we should all be very proud. It doesn't seem like there's much left for today, so I'll close us. Connie and I will be recording this meeting and posting it online. So if you have people who are interested in those ring trial results, or Andrew, we'll chat. If for some reason you don't want yours posted yet, we could take that out. But anyways, we'll have at least the ring trial results, because I know people are interested in that. We can point them to that recorded presentation. So big thank you to everyone. I have a nice list of things to get started on for the end of 2025, early 2026 here. So you'll be hearing from me and Connie. And yeah, just really appreciate everyone on the call. Thanks, and have a great rest of your day.
Thanks, everybody.
Thanks, everyone.
Bye.
Bye.